At this point, we're going to look at a lot of how living cells are put together. So to do that, we need to understand a bit about how everything is put together, and so we can approach that from the angle of looking at it chemically. So we'll talk about the chemistry of life, and just to go into a little bit about the nature of atoms and molecules and how they fit together to put things into perspective. Everything, all matter, what we would call, is made out of atoms, and that much you've probably heard. But atoms are then classified into different elements, and elements are all characteristic in terms of their size, the number of protons, electrons, and neutrons that they have that compose the atom. And so the protons of an atom are defined as having a positive charge, and they would be positioned in what we call the nucleus of the atom. So it's a different nucleus, of course, than what's in a cell. And then we have the electrons of an atom, and the electrons are zipping around the nucleus of the atom, and they are considered to have a negative charge. And then the neutrons have a neutral charge. Okay, so the neutrons and the protons are in the nucleus, what gives the atom most of its mass. So here's just a general picture of an atom, and you can see that this would be a carbon atom, that it has the protons and the neutrons here, the electrons zipping around it in these orbits or orbitals. And it's the interaction of the electrons in between atoms that will actually allow other molecules to be made. So with regards to the elements in any living organism, the most important and prominent elements that would be encountered are carbon, hydrogen, oxygen, nitrogen. And so most molecules are made out of primarily those four, and then also amounts of phosphorus and sulfur, as well as a lot of trace elements. And so what are some of the trace elements? These are some of the key salts that we might need, things like potassium, chloride, sodium, of course, and other minerals. So these are things like calcium and some of the metals that we recognize as important in our diet and for our health, such as iron, magnesium, zinc, etc. The role of a lot of these minerals or trace metals is that they can be important in helping our enzymes function properly. So to build on that a little bit, the atoms that make up everything are also held or bonded together by various attractions into bigger structures that we would call a molecule. So a molecule is composed of atoms that are sharing electrons, and that sharing of electrons is what we call a covalent bond. And then you can also have various chemicals put together by electrostatic forces. And electrostatic force is essentially the attraction between something that has a positive charge and something that has a negative charge. So that opposite attraction phenomenon can hold different compounds together. So one of the most important molecules to life, of course, is water. And so we could summarize water as being H2O. H2O means that it has two hydrogen atoms and one oxygen atom. And water is very important to life because it's what we would call a very effective solvent. It's very good at dissolving a lot of different substances. And the reason chemically that it's good at dissolving things is that it has a property that we call being polar. And that polarity is because when oxygen and hydrogen share electrons to bond them together, the oxygen pulls those electrons more strongly to it or around it. And so the concentration of those electrons around the oxygen results in the overall sharing and the overall molecule itself having a polarity, so that there's a negative pole and a positive pole. It's not quite the same as something being completely charged, so we call that being partially charged because the atoms are still just sharing the electrons. One hasn't taken an electron from the other. And so this polarity and being polar allows the water to interact with other polar things, and that's how it can dissolve certain substances like sugars, as well as allow water to have a cohesiveness when it's around other water molecules. And so one example is the surface tension of water. That attraction, which is what we actually call a hydrogen bond between water molecules, is what gives water its surface tension and what makes it a fairly concise substance. Of course, it has the fluidity too. Frequently measure, which is pH. So water is composed of 2 atoms of hydrogen and an atom of oxygen. Well, in any volume of water, these molecules are constantly coming apart and then reforming at a certain rate or with a certain level of equilibrium. And so that kind of reaction is such that the H2O can dissociate into a hydrogen ion and a hydroxyl ion. And the name ion is, just write that here. An ion is basically a fully charged molecule, ion. And that charge comes from the fact that it would have a actual different number of protons and a different number of protons and electrons in it. And so the hydrogen ion is actually just a single proton without an electron. The hydroxyl ion is formed because the oxygen essentially pulls an electron from the hydrogen, and so this unit, this package, the OH, has one more electron than it has protons, and so that's why it's given negative charge. So this is always happening in water, and in fact, if we would want to consider what we call the concentration of the free hydrogen ions in water, that concentration of H plus is what we define as its pH, and that is for any solution or any liquid, including beer. So when we have something that's an acid, let's say lactic acid is considered acidic because when it is combined or when it is added to a certain amount of water, then it's going to contribute additional hydrogen ions, additional H plus. And so that would, by increasing the concentration of H plus, affect the pH. How does the pH scale work? So let's understand this a little bit. So if we can recognize that the hydrogen ion concentration in any solution can be measured as a pH, the way we express that is what's called the negative log of the hydrogen ion concentration. And so what does the negative log mean? Well, let me just give an example. So, and to demonstrate this, we'll consider the difference between beer that's at a pH of 3 versus a beer that's at a pH of 4. And in fact, what we'll see is that the beer that's at a pH of 3 has 10 times greater amount of hydrogen ions compared to pH 4. The way that works is that beer that's at pH 4 has a hydrogen ion concentration of 0.0001, and the units we use to define that are called moles per liter, or molar. And that's just a expression that chemists use because it's able to directly relate these moles to the molecular weight of the compound. So if pH 4 has 0.0001 moles per liter of hydrogen ions, something that's a beer that's at pH 3 has 0.001 moles per liter hydrogen ions. And so that's where the tenfold or 10 times difference comes in. If we take the log of this number, 0.0001, the log of that is negative 4. If we take the log of 0.001, the log of that number is negative 3. So if the pH is the negative log of some hydrogen ion concentration, then the negative log of 0.001 is 3. The negative log of 0.0001 is 4. And so that's why we get our pHs. Now let's look at, we're going to move in to biomolecules, but we first have to start with a little bit of organic chemistry. So any molecules that are based on carbon and hydrogen are what we consider organic molecules. Organic molecules don't have to come from living organisms, but living organisms use organic molecules for pretty much everything that puts them together. So that's most chemical structures in living things, except for things like water and dissolved gases like oxygen or carbon dioxide and some salts. Most structural compounds in a living organism are based on organic molecules. And this includes all kinds of substances like our alcohols, sugars, amino acids, the proteins, essentially everything that's attributed to a living system is made up of organic molecules based on carbon and hydrogen, along with many other things, usually oxygen, maybe nitrogen. So a little bit about how one might see diagrams of organic molecules, since I know this is maybe new to a lot of you, to hopefully avoid confusion when we see or show these. It's important to understand some of the ways that they're drawn. So basically, when you're diagramming an organic molecule, and we'll use it as an example, glucose here, that it can be drawn in a number of different ways. So these 4 diagrams here are all different ones that are different versions of how we would show glucose. And glucose has a chemical formula of C6H12O6. That means it has 6 carbons, 12 hydrogens, and 6 oxygens. So this structure of it shows all the different atoms that make it up, including the hydrogens. And all the lines that are between them are, again, those covalent bonds, where it shows how they're stuck together by sharing electrons. And so this is one way of showing glucose, but most of the time for organic chemicals, since they're all based on carbon, the carbons are actually omitted from drawings in a way such as this diagram, or this one too. And so at every point or intersection or angle even, the person has to assume that it implies that there's a carbon there. So there would be a carbon here, and a carbon here, and a carbon here. Something else that is sometimes done as a shorthand is that even the hydrogens are omitted. And so in this diagram, we assume that there's a carbon here. And also because carbon forms 4 covalent bonds with the other atoms around it, if we assume there's a carbon here, then we have 1, 2, 3 covalent bonds, and to make up the 4th, it would have to be filled by hydrogen. Since that's its most common bonding partner, sometimes the hydrogens are not shown either. So these 2 pictures actually show the same things, just that this one omits the hydrogen. And then sometimes the angle that one is assumed to be viewing the structure is changed too. So this is actually a picture that's the same as this. It's just that it's assumed you're kind of looking at it from more of an oblique angle, because the carbons of sugars like glucose form themselves into ring structures. And of course a lot of work has gone into by chemists to kind of come up with these models for these kinds of diagrams, because it's much too small to be seen with a microscope. No microscope could see the actual atoms that make up something like glucose. So we have to just think about it with models. So with that, and hopefully you'll get used to a little bit of looking at some of these diagrams, because I show some of the others when we get to the metabolism section, we should look at how some organic molecules are built. And so a lot of times they're built of various pieces, kind of like Lego models, and these pieces can be assembled in a large variety of different ways, and really limitless. And so just to get an idea of what some of the types of pieces that are important in organic and biomolecules are, we'll look at some of them here. So the first one, obviously very important to beer, is alcohol. and any kind of a compound that is considered an alcohol, and ethanol is the alcohol that's produced in beer, but there are many, many, many other kinds of chemicals that we would consider to be an alcohol. Even, for instance, rubbing alcohol, isopropyl alcohol, is just one example of many others. Any alcohol is going to be defined by having a hydroxyl group on it, and a hydroxyl group is OH. And so in this example here of ethanol, we can see the OH that is on the end of this chain of carbons. Most organic molecules are built as either a chain or a ring of carbon atoms put together with some of these different appendages. And so OHs make something an alcohol. Now, we'll also see that something like sugars have a lot of OHs on them. But if we just have a carbon chain with an OH, it's an alcohol. Now, when we have what's called an amine, and an amine is what is something that's inferred to contain nitrogen. So in this picture, which is an amino acid, the amine group is this, the nitrogen with two hydrogens on it. And it's just that particular group that is the amine. The rest of this is other parts of the molecule. We'll hear a lot about esters. Esters can have a lot of different forms, but an ester in general is some chemical or organic compound that has a functional group that is something like this, where we have a carbon with one oxygen that's sharing two pairs of electrons, and then to the side of the carbon another oxygen. These R's represent extensions of the rest of the molecule, so you would have more carbons kind of going on one side and the other. So an example is the compound methyl acetate. You can see the ester component of that right here, where we have to assume that there's a carbon here, so this is drawn with that shorthand. There's a carbon bonded to an oxygen with a double bond and another oxygen with a single bond. So that structure makes it an ester. So we'll look at the importance of esters as a type of compound that can affect the flavor in a little bit, or actually in the next lecture. Other kinds of important functional groups that are on organic molecules include what's called a carboxyl group, or what makes something a carboxylic or organic acid. And various acids that can be produced by yeast are also something that can affect the flavor as well. So what makes something an organic acid, such as acetic acid or lactic acid, two of the biggest ones of concern in brewing? It's this carboxylic or carboxyl group here that is typified by, at the end of the carbon chain, of course this is a very short chain here, just two carbons, but at one end it has a carbon, again double bonded to an oxygen, and an OH. Now, the thing about these carboxylic acids is that when they're actually in a water-based environment, that the hydrogen is often lost from the structure, the hydrogen that's here on the hydroxyl. And so what will happen is that the oxygens here produce such a strong pull on the electrons that it'll strip or remove the electron that it's sharing away from this hydrogen and release the hydrogen as just a free proton. Remember, proton's the other part of the nucleus of an atom. And so what you would see is that after that occurs, one might draw or depict something like lactic acid, or at the end of this structure here, I'll just draw the end of it as having a carbon and the oxygen double bonded to it, and then here we would have the other oxygen, and because it has pulled that electron away from the hydrogen and that hydrogen left, it's kind of short one proton now, and so we would give that a negative charge. I guess to round it out, you'd have the other carbons over here and the hydrogens that go on them. Okay, so this is considered what makes it an organic acid. And the key thing is that it's also released that hydrogen ion now. So we'd say that it's like this over here. And so that would contribute those to the solution. And what does that do to the pH? Well, when you add those hydrogen ions to the solution, then that increases their concentration, and that actually lowers the pH, because the pH is the negative log. So it's an inverse relationship. And then a couple others to note are those that are called thiols. So a thiol is something that has a sulfur, and it's also what we call a sulfhydryl, sulfur and a hydrogen. Well, this would be connected to a larger molecule, but in general, anything that has sulfur or is a thiol is one that can often contribute a noticeable or pungent smell. A lot of our real, kind of atrocious smelling or unpleasant smelling or strong smelling things, that smell is attributed to sulfur or to thiols. So, for example, hydrogen sulfide, the rotten egg smell, or mercaptans that we'll cover that kind of are what give natural gas its smell. Even burning hair or other tissues that have keratin in them releases the sulfur from that, or matches when they're burned, and you smell that sulfur. And then lastly, phenols. So phenols are a ring-based structure made up of carbons, and for the carbons that are in these ring-based structures, that there are also a number of double bonds where the carbons between sharing the electrons between adjacent carbons, you have two pairs of electrons being shared. So phenols also have characteristic flavors and smells that we'll discuss in the next unit on metabolism. So let's now put this together into the things that make up living cells. And so we can get into the biochemicals now. So we can look at four main general types of biological molecules, large biological molecules. And those are the polysaccharides, which are made up of chains of sugars, and the proteins that are made up of chains of amino acids, or nucleic acids, and this is how one defines DNA, which is a chain of components called nucleotides, and then structures that are known as lipids. And lipids have a bit more variability to the way they can be put together, but the different kinds of lipids that are important for living cells are fats and oils, substances called sterols, and phospholipids. And they have some important roles. So sugars, and when we have big chains of sugars as polysaccharides, have some important roles. So simple sugars have a very important role, as you might guess, in energy storage. So energy from sugars, or sugars provide a very important source of potential energy that cells can use. But when we put a lot of sugars together, in certain ways, they can make polysaccharides, and polysaccharides have some very important structural functions that we'll see when it comes to especially the cell wall of yeast and bacteria. Now, proteins have a variety of different roles, and some of those most important ones is that they can form structural molecules, but also proteins are what makes up enzymes, and enzymes are very critical to the functioning of all forms of life, because it is enzymes that allow the chemical reactions that make life happen to take place, and to take place in an efficient way. So enzymes are kind of the workhorses of the cell that make everything move and work. But also we have proteins that are important for transport of substances into and out of cells and within cells. And then proteins can also provide signals between cells too by being transmitted. So if you look at an example maybe from humans, insulin is a signaling molecule that's a protein. Nucleic acids are important for transferring and for storing genetic information that encodes for how to make the proteins. The proteins as enzymes are basically what put together things like sugars and then when we get to the lipids. Lipids are also primarily a structural role as well as important roles in energy storage, and also for sending cell signals as hormones. So some of the other signaling molecules include, for instance, testosterone or estrogen in humans, not in microorganisms, but hormones are made as steroids, which are a type of lipid. So if we look at the first group there, the sugars and polysaccharides, what are these? Well, we can define these as carbohydrates. So it's what makes something a carbohydrate. our general carbohydrates are based on the formula that we can show as a CH2O and then N as a variable number of multiples of that that formula. And so that's partly where you can look at that and see why they're called carbohydrates. So carbo, because there's a carbon there, and hydrates because there's a water there, H2O. So carbohydrates means that we have carbons with basically waters. As sugars in water solution, these carbohydrate molecules take on a ring-like structure. And so if you think back to glucose that I was showing the example of, those are formed as a ring. And so in fact glucose is the main sugar that we actually consider. It's the most kind of elemental or essential sugar that is important for life. Many of the other sugars that we have, including maltose or sucrose, sucrose is table sugar, and these are primarily based on glucose. And then other glucose can be modified into different shapes or take on different extensions that oftentimes are forming different structures. So glucose is kind of the main sugar to remember. If we want to look at maltose, of course, very important to brewing, maltose is actually what we call a disaccharide, so it's 2 units of glucose. If we just have glucose, that would be a monosaccharide. And glucose or other sugars can be linked into very, very long chains to make what we call polysaccharides, because poly is a word that means many, and saccharide is a word root that refers to sugars. So when we string long chains of glucose together, in some forms they can create big carbohydrate polysaccharides that are useful for energy storage. So very important to plants is starch. So plants form the sugar that they store into starch. And in animals, as well as in yeast, we form sugars into big long chains that we call glycogen. So in a human, the glycogen is stored in the liver and muscles to be used for rapid energy. In a yeast cell, glycogen can also be stored. But we also want to think about the importance of starch as it relates to brewing. And so, of course, what's the main source of sugar for brewing? It is the malt that we use, the barley or other grains. And so those grains are a form of starch that is coming from plants, and so they're basically a chain or long chains of glucose. Other ways that polysaccharides are important is that the yeast cell wall is made out of a polysaccharide largely made out of what's called chitin. And chitin is long chains of sugars that are derived from glucose, but they also have nitrogen extensions. And the name of this sugar is called N-acetylglucosamine. And basically, if you look at the glucose part of it, it shows how there's and this is again a different angle to looking at the glucose shape, why it looks kind of crooked like that, but that's where the ring shape, if you were to kind of rotate yourself above it, might look like this. So this is showing glucoses put together, and we also have some nitrogens on there, which is why it's called glucosamine. And the acetyl refers to some other appendages on it with the carbon here. So that is what makes up chitin, basically a derivation of glucose. And plant cell walls are made out of long chains of glucose called cellulose. So cellulose is essentially what forms fiber that we would think of as being in our diet. And so it's simply just glucose, but it's in a different arrangement, a different form than what forms starch. So in fact, the glucose chains in starch are very digestible. The glucose chains in cellulose are quite indigestible, and that has its basis primarily in the number and different types of enzymes that can break it down. So we'll talk a bit more about the breakdown of sugars and carbohydrates in the metabolism unit. Proteins are chains of smaller pieces called amino acids. And so there are 20 different amino acids, and they all have different properties. And the way that they're put together gives proteins their variety of the different shapes that they have. And the different shapes that they have is primarily what gives them the different roles or functions that they can play. So one of the main roles that proteins have is as an enzyme. So we'll talk a bit about some of the important enzymes in yeast and in brewing. But enzymes are a catalyst. So a catalyst is something that makes a chemical reaction go faster. And so they can actually essentially trigger, by being present, they can trigger chemical reactions when the enzyme is present. So enzymes are a type of protein, and proteins are made out of amino acids. The information in order to make all these protein enzymes is what comes, or is what's stored on the DNA. So the DNA information is translated into making proteins, and those enzymes, those protein enzymes, then become the tools and the little machines that, by doing chemical reactions, are what can form all the other parts of the cell and do all the other functions. Enzymes themselves are very specific. They're highly specific for particular reactions. They don't necessarily just perform one exact chemical reaction, but they usually have still a high specificity for the chemicals that they work with. And that specificity and as well as their efficiency and their ability to work comes from their particular structure, their shape. And one of the ways that the shape of an enzyme can be affected or changed is by environmental conditions of what's around it. So they're very highly dependent on these types of conditions for their particular structure. So those conditions are things like temperature, which is very important, the pH, as well as other ions, other charged atoms in solution, or charged molecules in solution. So another ion might be like sodium ions or chloride ions, salts. And of course the pH is the hydrogen ions. And so these can be important for the exact structure of a protein. If they're off slightly for what that enzyme is adapted to, then that can dramatically affect its reaction rate or make it not work. So this large variety in different shapes is what gives them their specificity. And also it may come up that the reactions that enzymes can perform a lot of times are reversible. They can go in two different directions. They may break something apart as well as put those two things back together, depending on the reaction or and the concentrations of both the reactants and the products. So there's a lot of very important enzymes in brewing. So just for a review of that, you should look over in the book Yeast by White on pages 30 to 34 and review some of those key enzymes. One of them, and one of the most important ones in brewing, is the enzyme called alcohol dehydrogenase. So I wanted to highlight that a little bit. This is basically what makes yeast fermentation work. And so a lot of study, of course, has gone into this. The way that biochemists can now, with advanced techniques, do studies is to actually crystallize the enzyme proteins, and then using X-ray imaging, they can get an idea of its actual molecular shape. Along with understanding the gene sequence and the amino acid makeup that goes into the enzymes, they can model how the atoms would interact with each other and come up with models of their form. And so these images are showing the structure of various renditions of alcohol dehydrogenase. And so in this structure you can actually see models of where the amino acids are. Of course, they're just a series of different, basically organic chemical forms, so you have the hydrogens, and you have the carbons around there. You can pick out maybe a few rings, the places where it's red or where there's some oxygens, places where it's darker blue or where there's some nitrogens. And then one could also do a rendition that shows the overall path that the chain of amino acids takes, and the the different structural characteristics that it has. And so we can see, and in fact, that alcohol dehydrogenase is made up of two units that fit together. So this enzyme is what gives us carbon dioxide and ethanol during fermentation. A bit on lipids. So lipids are very important in cells too. Lipids include our fats as well as steroids and the forms that make up the membrane of cells, which are called phospholipids. So these are defined and characteristic in being what we call hydrophobic. And so something that's hydrophobic means that it doesn't interact well in water, or it doesn't mix well in water. It doesn't dissolve in water, but it does. It is soluble, or it does mix in fats or oils. And so hydrophobic things are oil soluble, although they may still have some hydrophilic parts. Something that's hydrophilic is able to interact with water. And so the main types of lipids include fats, and fats also include oils. So you might think of fat as, bacon grease, and oil might be just like vegetable oil. These are what you call your triglycerides. They're called triglycerides because they are composed of a glycerol molecule. Glycerol is a type of alcohol that's connected with three extensions called fatty acids. So a fatty acid is essentially a long chain of carbons and hydrogens. And so it's kind of like a hydrocarbon in that way. So the structure of a fatty acid also has some variability. And if we look at this picture of a fat, we see here this part's the glycerol, and these parts are the fatty acids here. Now the fatty acids can either be saturated, which means that all the carbons going down the chain of the fatty acid are saturated with hydrogens. Or a fatty acid can be unsaturated, and if it's unsaturated, that means that in some places where it could have a hydrogen, it doesn't have a hydrogen and it has a double bond to share those electrons between two carbons. And so this double bond in a fatty acid chain makes it unsaturated. Unsaturated fatty acids and when they're composed in fats, then when a fat is made up of more unsaturated fatty acids, it's more fluid. So that's what makes oils liquid and fats like bacon grease or lard more solid, is that bacon grease and lard have mostly all saturated fats or saturated fatty acids, and something like canola oil is made up of a lot more unsaturated fatty acids that have these and the reason being that the fatty acid, when it has the double bond, it gets like a bend in its shape, and that prevents them from packing together so closely. And then lastly, we have lipids that are sterols, and types of sterols include cholesterol. And cholesterol is shown here. Cholesterol and other steroids or sterols. A steroid has these joined rings structure. A sterol is a steroid that also has an alcohol or an OH on it. So these sterols, such as cholesterol and ergosterol that yeast produce, is similar in a structure being like this. This whole part of it is all just carbons and hydrogens, and then it has one hydroxyl group, or OH, on an end. The significance of sterols is partly why cells or yeast in fermentation need oxygen. It's the incorporation of oxygen onto the steroid frame that is what allows the production of sterols that allow it to make things like ergosterol that are important in membrane stability. So phospholipids, along with the sterols, are what put together the cell membrane, and the membrane is the enclosure of the cell. And so when we look at phospholipids, they're kind of similar to a triglyceride that was shown on the previous page, but in this case we have still the glycerol component, but this time we have two fatty acids, and instead of the third fatty acid we have a charged phosphate group. That's why it's called a phospholipid. And when I say charged, it's because it has some negative charges on it. So it's got an unequal number. It has more electrons than it has protons. And so that charged aspect of that one side makes that side have an affinity to water, because water is partially charged, and so it can interact with the charged side of a phospholipid. The fatty acid side of a phospholipid has only just the carbons and hydrogens, and that part is hydrophobic. So when you have a membrane, when you have a whole bunch of these phospholipids together, they will associate such that if there's water, they'll form what's called a lipid bilayer. And this little figure A here shows a lipid bilayer with the fatty acids all orienting together because they're hydrophobic, and then the head of it that has the phosphates all orienting outwards towards water on either side of it. Cells have water both inside and outside. But you have this membrane structure that can be formed. So the cell membrane is a phospholipid bilayer. And like the fats, it can have varying degrees of saturation on those fatty acids. So those phospholipids that have more unsaturated fatty acids tend to make membranes that are more fluid. It's because of that, again, that bent shape that occurs from the double bond on the unsaturated fatty acids. In addition to the phospholipids that make up a membrane, there's incorporated a lot of sterols, such as ergosterol in yeast, as well as a lot of important proteins that have roles in transport. And the sterols themselves are important to help maintain somewhat of a strength, a rigidity, and an integrity to the phospholipid membrane.